Well, hello everyone. My name is Alexander Bartelt. I am the professor of cardiovascular metabolism at the LMU in Munich, Germany. And it's a great pleasure for me to be here today and present to you something old, something new, and something that we don't yet understand about brown adipose tissue. And I have to give special credit to Phil White, who is organizing this and invited me to speak here to you today. And I think it always requires a lot of courage and a lot of work for leaders like Phil to bring us together and talk about our work and exchange thoughts so that the science can move forward in these difficult times where we have to live pretty much in isolation. So I'm best known for my love of brown fat, so I chose to talk today about brown fat, but nevertheless, I also want to highlight the work that we do in our lab. We are not only focused on brown fat, we anticipate that metabolic disease, all the way from disturbances of energy balance, what happens when we gain weight, how are adipocytes kept healthy, and then ultimately how does that lead to cardiovascular diseases and diabetes, that there are ancient mechanisms that play an important role along the way. And this is the main topic of our lab, metabolic adaptation. I'm very thankful to the funding that we received from the European Research Council, the German Research Foundation. And particularly this project today is also a joint collaboration with the lab of Stefan Herzig at the Helmholtz Center Munich, where I'm also holding a principal investigators affiliation, and we actually share a couple of people and projects that we coordinate together. And this is particularly supported by the German Center for Cardiovascular Research. So metabolic adaptation is a very important topic, and there are environmental challenges that stimulate our metabolism and requires that our body, our cells adapt to different conditions. And one of these conditions that is very critical for our body as homeotherm mammals is cold. And this is a picture I've taken during my postdoc in Boston, and you can see here, it's just a regular winter day in New England. There's a blizzard, there's a lot of snow, freezing temperatures, and that's very challenging for the human body. And this metabolic adaptation to cold is a process that can be described in this diagram. And this is, I have to point out, for a mouse, a rodent. And you can see here that these animals, there is a thermoneutral zone where the basal metabolic rate is already enough to produce so much heat that the body temperature can be maintained in a certain window. And once the animal leaves this thermoneutral zone, then it needs to recruit extra metabolism for heat production, and this then also leads to increased oxygen consumption. And it's important to point out that for rodents, particularly for mice, about 1 degree of ambient temperature needs to correspond to 8 to 10% increase in energy expenditure. So that obviously is a very large margin for the mouse to be able to adapt to. And one of the mechanisms mice have, and actually all mammals except maybe for pigs and horses, is a tissue called brown adipose tissue. So what is this? This is a tissue that, unlike white fat cells, contains cells that are thermogenic. They are both, they look different. They have a multilocular lipid droplet structure and contain large amounts of mitochondria, hence the color. So the iron, the more mitochondria, then makes these cells look more brownish, yet they are still a fat tissue. And the cells are also much smaller than those lipid-laden white fat cells, and they possess special mechanisms and then are usually classified as being UCP1 positive. So what is UCP1? UCP1 is the uncoupling protein 1, which sits here in the membrane of the mitochondria. And thermogenic fat cells, they are activated the most powerful stimulus for their activation is obviously cold, which is then sensed through the skin, translated through the brain, and then this leads to activation of the sympathetic nervous system. And the norepinephrine is a powerful neurotransmitter that directly activates these cells. And what then happens is that the cells break down their stores of lipid droplets and glycogen to generate energy derivatives, which in most cells would lead to the generation of ATP. But brown fat cells, they have a special trick. They have actually very low rates of ATP production, and most of the energy doesn't go into ATP. It's actually dissipated into heat, and this heat is then transported to the periphery of the body through the blood to keep the animal warm. And obviously, this is a process that needs to be fueled by stores that are available within the cell. And once those run low, the cells are very actively recruiting the nutrients from the outside to fuel this energy demanding process. Brown fat is not new. You could almost call it ancient. It has been discovered in the 16th century by a Swiss anatomist who described it as being neither fat nor flesh. And we know that humans contain multiple depots of brown fat, and this is shown here in this review. We can see that you can find human brown fat here in the supraclavicular area, in the cervical region of the neck, and then paravertebral along the spine. And there's also a shift that is very different also from rodents. We know that children, infants carry much more brown fat than adults, and the mechanisms behind this transition to adult human brown fat are not very well understood until this point. Why are we excited about brown fat? And I think this is sometimes sold as there was a rediscovery of brown fat, even though some people in the field always appreciated that the humans have significant amount of brown fats. And for many, many years, these patterns that appeared in the PET-CT scanning of cancer patients were dependent on seasonal, were dependent on room temperature, and then these turned out to be actually hotspots of glucose uptake by brown fat. And there were a couple of papers that then showed that at colder temperatures, the glucose uptake, the tracer uptake into these areas increased, and that was dependent on the temperature. So when the subjects were held at warmer temperatures, these patterns did not show up. And what was also very interesting for the metabolic disease field at that time was that obese people, people with a higher BMI or older people, generally showed less uptake of these tracers into brown fat, indicating that obese people might have less brown fat, and potentially that having less brown fat would also enhance their obesity. Some of this has been refuted. Nevertheless, I can tell you that the overall notion is now brown fat makes a difference, even though there's arguing over how much actually brown fat makes a difference. But at least, for example, in this paper, you can see that people that have detectable brown fat, they increase with cold exposure roughly 200 calories, the cold-induced thermogenesis, which is significantly more than people that don't have brown fat. I will get to this in the end. And brown fat in obese people can also be trained. So it's not necessarily only that they don't have brown fat, more likely is that they're not using it, or it might become dysfunctional. And you can also train brown fat in people that are overweight. You can see here in the study, 10 days of 15 degrees cold exposure led to the re-emergence of these brown fat speckles, indicating increased activity. There's also a way to pharmacologically activate brown fat in humans, and this is a picture that was taken before the injection of an FDA-approved drug called Miabergon, and injecting this drug, albeit at very high doses, then led to the activation of intrinsic brown fat. What is the metabolic benefit of activating your brown fat? And there's accumulating evidence that Brown fat not only does thermogenesis in humans, it also impacts our systemic metabolism and metabolic health. These are two studies that appeared in JCI and are from last year, where the authors showed that when you use myabragon, it improves glucose homeostasis in obese humans, and also has beneficial impact on your plasma lipid profile, HDL cholesterol. And some of this actually has been mechanistically also elucidated in mouse models. We know which genes are important, and this was, if you like, the translational confirmation of a lot of the things that we've been studying in mice for a long time. Also this year, there was a great study where a lot of PET CTs of a lot of patients have been studied, and you can definitely see that here the correlation is very clear. People that have brown fat, they are healthier, they have a lower risk of cardiometabolic disease clusters, they have improved glucose and improved plasma triglycerides. It's actually interesting that people that have a higher BMI are better protected than people that have a lower BMI. And that does make a lot of sense. So the conclusion of the human data so far is that really you should adopt a thermogenic lifestyle and train your brown fat, because that's good for you. Just like Leonardo DiCaprio here in the movie, where he was straining through the wilderness and probably came back with a lot of brown fat after his excursions. So human brown fat is real, but obviously one major question is, how does it work and how can we study it? And that obviously is very complicated, because it's difficult to find brown fat. These have to be PET CT guided biopsies, and then we can mostly study the cells or study the tissue ex vivo. And most of the knowledge that we have is about acute physiology that was done with tracers. And the molecular signatures that we have of brown fat are mostly derived from ex vivo studies. And I think a major question also for the fields is then always, is the rodent a good model for studying human brown fat? How much of the physiology from mice can we translate into humans? And are actually the molecular mechanisms conserved between the two species? And one major difference, which is very obvious, is I said that this diagram here of metabolic adaptation to cold is for rodents. And it is obvious that for us, a thermoneutral set point is probably at a very different temperature than for a mouse, because of our different surface to volume ratio and overall different and slower metabolism also. So in the mouse, you see that a housing temperature leads to very different morphology of the brown fat and also different functionality. In humans, we know that a standard house rodent mouse model here, interscapular depot, and this is a paper from Jasper de Jong that came out 2 years ago. And you can see here that in a regular mouse, brown fat looks very nice, very brown, whereas human brown fat is kind of like somewhat brownish, but also a little whitish. And then what they did through changing housing temperature of the mice and a lot of phenotypic analysis, what they put forth is that the humanized condition for a mouse is when you actually put them at thermoneutrality. And then when the brown fat becomes lipid-laden, also the signatures between mouse and humans become more similar, and that would be a better approach actually to mimic some of the physiology of human brown fat in mice much better. Now, what kind of brown fat do humans actually have? And I think there's also still some disagreement over here, but I'm usually relatively neutral to this. I think it depends where you look. This has also been put forth by some of the other experts in this field, and you can find all sorts of different brown fat phenotypes in humans, both classical brown, so-called bright or beige or white fat cells. It really depends on which depot you're looking at. And some of the markers that are put forth as being human brown fat markers are actually, in the end, only much more of a marker for the depot, the anatomic location, whether it's a supraclavicular fat area or deep cervical. And this is also, again, work by Jasper de Jong, who shows that these markers are not really corresponding to functionality, but they're much more corresponding to the location of the fat tissue. And I think that's also important when we talk about what is a good marker for brown fat, and is there actually a good marker for human brown fat? In the end, nevertheless, human brown fat is different from mouse brown fat. And what I'm going to talk to you a little bit more now is that it's not necessarily in the genes, but in the intergenic regions. And this is a very nice overview, a study on the evolution of long non-coding RNAs that was published a couple of years ago. And here on the left, you see the graphical abstract that the landscape of long non-coding RNAs obviously is very different in the animal kingdom, and that throughout evolution towards the development of humans, there has been significant remodeling of the non-coding genome. And here then on the right-hand side, you see that there's a large proportion of genes or of the genome actually that are non-coding in humans, much different than two lower species, let's say like reptiles. And this also has a functional relevance. This is a paper from last year from the group of Søren Nielsen, also a large collaboration, which is always required when you study human adipose tissue, because it's not so easy. And what they found here in the sequencing, so they compared supraclavicular and abdominal subcu fat versus other fat cells that were treated with norepinephrine, one of the primary activator of thermogenic fat cells, and that they found that there was a large array of long non-coding RNAs that were regulated under these conditions. And they went on to show that either with endogenous overexpression or with loss of function studies, that this long non-coding RNA actually localizes to the interface between mitochondria and lipid droplets. So it's probably not regulating gene transcription at that level, but it's rather really at the post-translational regulation, a scaffolding RNA that regulates lipolysis. And this, of course, was very interesting. And at the same time, the Maud Giroux, who was at that time working with Stefan Herzig, was also interested in the question and what is new about human adipocytes. And when the paper came out last year, actually, we were already working on the project that I'm going to tell you about now. And it was very comforting to us because we also had discovered this long non-coding RNA in our screens. So the work that I'm going to show you today is unpublished. It's in preparation. I think we've gotten pretty far with our understanding what's going on, but as always, there are a couple of points that are still on our to-do list to really make this a full story. And it's, again, a joint collaboration. So Maud is a postdoc in the lab who came from France, where she already used to work on a cell line called hMADS, which is a mesenchymal human stem cell line, which has high plasticity and can be both brown and more bright or beige in the differentiation protocol, but I'll get to that. And she started doing her postdoc with Stefan Herzig at the Institute for Diabetes and Cancer at the hMADS Diabetes Center here in Munich. And now she is a postdoc in our lab at the Campus Großhadern at the LMU. So, and this work she performed together in collaboration with Stefan Kotzi, who is a MD student in the lab. And what they were actually doing was in the beginning to understand, okay, so if we put together the knowledge that we have about tissue, so human tissues, brown versus white, then human primary cells that were treated with control or norepinephrine, just like in the previous paper that I showed you, and compare this to the cell line, the hMADS, that were treated with forskolin to stimulate the CAMP signal. When you put all these data together, what comes really out of it, and what can we make out of this knowledge? And in the end, she identified 25 hits, one of them being the LncRNA that was published last year already. And the one that she has been focusing on ever since is called AATBC. It stands for apoptosis-associated transcript in bladder cancer. I don't have the time to go into much more detail what is already known about it, but I can tell you at that time, nothing was known about its role in adipocytes or in thermogenesis. So again, how do you study human brown fat? It's difficult to study human brown fat in an in vivo system, like in subjects. So we went on to use this hMADS cell line, which, like I mentioned before, depending on how much rosiglitazone you add to, you can really make them nice, bright or beige with relatively large amounts of UCP1 and uncoupling, or keep them in a much more wide format, with withdrawal of rosiglitazone towards the later stage of the differentiation. And then she used two approaches here to manipulate the function of AATBC. One was by knocking it down in this bright differentiation protocol. And the reasoning behind this, as you will see, is that the bright cells have a higher amount of this long non-coding RNA, whereas in the white fat cells, which have less of this non-coding RNA, you overexpress it with a virus, and then you have a much stronger gain of function set of experiments. So, like I said, it's higher expressed in the thermogenic cells, and by RNA-scope, we actually found that most of the long non-coding RNA is located in the nucleus. And what was interesting to see in the beginning was that when you knock down AATBC, you do get less UCP1, both on RNA and protein levels in these cells. And that obviously is already very interesting to start with, as UCP1, next to some other alternative thermogenic mechanisms, obviously is the main mediator of non-shivering thermogenesis. And we also did seahorse experiments, and you can see here both with gain and loss of function, you manipulate the oxygen consumption rate and the maximum capacity of these cells, indicating that there is an innate mitochondrial difference when you manipulate AATBC. And this could also be confirmed by mitochondrial DNA, which is less in the knockdown cells, and there's also more mitochondrial branching in the knockdown cells. So that would indicate that the mitochondrial network in these cells is somewhat different, which would then lead to a different thermogenic capacity. So we then looked into this further, and by doing TOM20 staining and some imaging analysis, we actually found that when you knock down AATBC, there is a difference in mitochondrial length. There's increased junctions and increased branching. So the loss of AATBC actually increases mitochondrial connections. And conversely, when you overexpress AATBC in the more whitish fat cells, then you see the exact opposite effect. And this then complementarily shows that the gain of function actually decreases mitochondrial connections. This also goes hand in hand with alterations in mitochondrial dynamics, at least shown here for Western blotting of some of the known markers, which are known to influence mitochondrial dynamics in thermogenic fat cells like OPA1, mitofusin, and DRP1. And complementary to the imaging, you can see here that manipulation of AATBC impacts mitochondrial dynamics in a complementary approach. So what is happening, as AATBC is a nuclear long non-coding RNA, obviously one of the big questions we had is it maybe a gene regulatory mechanism, and if so, what are the gene clusters that are being altered upon loss of AATBC? And so we went to RNA sequencing to see what are the global changes in the loss of function of AATBC. And what you can find here, there were discrete clusters that were very much in line with the previous data, where we could see that the response to oxidative stress, response to oxygen levels, cellular response to oxidative. So a lot of clusters that would directly implicate AATBC in mitochondrial function. So this brings me now almost to the end of this small aspect of our work. The long non-coding RNA AATBC we find to be located in the nucleus. We find it to be increased by adrenergic signaling. We think that there is a nuclear interaction, and I pointed out here some of the major questions we're still trying to address that there is a link to the transcriptional response to oxidative stress. So we're focusing now on this discrete subset of transcription factors that are important for this response. And we can then see that depending on the transcriptional response, there are alterations in ROS levels, and that along the ways changes the mitochondrial dynamics, mitochondrial network, and thermogenic function of adipocytes. Obviously, another very important question that we will have is, can we translate this human function, the human cells, and as this is a human exclusive long non-coding RNA, we can't study it in the mouse, but can we translate at least some of the biology that we discover from humans back to the mouse to study its physiology and the mechanisms in more detail? So this is the work by Maud and Stefan, and I'm grateful to have them in the lab. They're extremely bright and very talented, hardworking students. But of course, all this would have not been possible without the help of more people. And this is the big acknowledgement here. So it's a joint collaboration between the HADSIG lab and the Bartelt Lab. And particularly, Maud has had great help from Marcel Scheiderer, from Manuel Losanza, and Jung Kwang from Anja Zeiger's lab, who helped with the image deconvolution. And how can you study humans? You need access to human samples, human biomaterial, and we were very thankful that we were able to collaborate here with Kirsi Virtanen and Matthias Blüher, also with Daniel Teves and Pamela Isha Povoscik to supply us with more white fat samples. And I mentioned earlier that the lab of Søren Nielsen has published already work on the long non-coding RNAs, and we're now actually teaming up and join forces to understand the phenotypes of long non-coding RNAs in non-shivering thermogenesis a lot more better. And of course, in the end, Maud was supported during a postdoc by Alexander von Humboldt Fellowship, which enables foreigners to come to Germany for a limited amount of time and work on a top-notch project. And obviously, we have to thank also some other funding sources like ERC and others who have helped create a prosperous environment in which this project was able to be performed. And with this, I'm at the end of my presentation. I thank you all for zooming in and your attention. And if you have more questions, please direct them to me personally via email or Twitter. You will find the details in this talk. Thank you very much. Bye-bye.